And so our last speaker, it's my pleasure to introduce Stella de Haan from Université de Liège. And she's going to talk to us about circadian sleep regulation, cognition, and brain structural function integrity in healthy older adults. So we go to the other end of the lifespan.
Hello, everyone. As my PI couldn't be here, I'll present our group's work today. I have no disclosures. So when we age, as we all know, our brain structure and functioning changes. And while it's well established that cognitive functions, such as memory and attention, tend to decline with age, this decline is far from uniform. So we can see that many people their cognitive performance tends to decline a lot with age, but there are some people that keep a very high cognitive performance well into old age. And understanding the factors that drive these individual changes is essential if we want to promote healthy cognitive aging across the population. And one factor that has received growing attention in recent years is sleep. So as we age, our sleep patterns change. Older adults often have earlier bed and wake-up times, have reduced nighttime sleep duration, have reduced slow-wave sleep, and a reduced sleep consolidation over the 24-hour cycle with increased fragmentation. And these changes may not just be a consequence of aging, but potentially could play a role in shaping how our cognitive abilities evolve over time. Sleep is increasingly being suggested as a key protective factor in healthy aging, and it is involved in processes critical for cognitive function, such as memory consolidation, metabolic waste clearance in the brain, and emotion regulation. While it is well reported that the structure and timing of sleep changes across lifespan, a consensus on the underlying mechanisms has not yet been reached. And to better understand the link between sleep and aging, we need to look deeper into sleep regulation processes. And to do so, we can use modeling. This is probably all known to you, but for the past 40 years, the two-process model has been heavily used in sleep research. In this model, the alternation between sleep and wake states are modeled by two physiologically distinct quantifiable processes. The first one is the homeostatic drive S, here that builds up over our time awake and declines during our time asleep. And the other one is the circadian timing system, C, a biological clock that provides us with an almost 24-hour cycle of low and high sleep propensity, promoting wake during the biological day and promoting sleep during the night. And while this model successfully can simulate the timing and intensity of sleep, it does not provide us with insights into biological mechanisms. For that, we need to move to biophysiological models, such as the arousal dynamics model. This is a version of the Phillips-Robinson model, and as you can see in the simplified version shown here, at the core, the model consists of the wake-promoting neuronal populations and the sleep-promoting neuronal populations, and they have a mutual inhibitory action such that they behave as a flip-flop switch. Only one can be active at the same time. And as you can see, the homeostatic and the circadian drive input on this flip-flop switch. So this model has been used to improve our understanding of sleep and its mechanisms. Skeldon and colleagues used a different version of the Phillips-Robinson model to show that age-related changes may be best explained by an overall reduction in sleep need and a concomitant reduction of circadian amplitude. So what this means in the model, that we can have a reduction of the circadian coupling to the sleep promotion, or a reduction in the homeostatic buildup, or a reduction in the homeostatic coupling to the sleep promoting populations. If we want to assess the circadian amplitude in humans, we can use a multiple nap protocol, which is shown here. This allows us to assess sleep ability over the circadian cycle under constant homeostatic sleep pressure. So participants come to the lab, they undergo an 8-hour baseline sleep, and then they have a 40-hour protocol, where in this case they have 10 times an 18-minute nap opportunity. And during this protocol, we record polysomnography, and they have constant light and body positioning. Using such protocols, we can indeed detect age-related changes. So in this cohort of 30 older and younger adults, we observe here that this is shown the sleep efficiency over the protocol, that we can see there's a clear circadian modulation with more sleep during the biological night. And we can also see that for the younger, they have a higher sleep efficiency over almost all naps, except for the wake maintenance zone. If we then look at the circadian amplitude, which is basically the difference between average nighttime sleep efficiency and daytime sleep efficiency, we see that the circadian amplitude in younger is higher than in the older. If we look at REM sleep expression, we also see that the strong circadian modulation and the younger have a higher REM sleep expression than the older. And also what is important to note here is that also the circadian modulation of the REM sleep, the difference between day and nighttime sleep, REM sleep in younger is higher than in the older. And REM sleep is increasingly being suggested as a discriminant sleep stage for age-related changes. As REM expression is under strong circadian control and circadian dysregulation increases with age, it is interesting to look at the association between brain aging and REM regulation. So to do this, we also collected MRI imaging for the same participants, and we observed a significant association between our measures of circadian amplitude in the aged and sulcus depth. So we observed an overall age-related reduction in REM sleep throughout the circadian cycle, and in the older, we observed that the higher day-night differences in REM sleep were associated with an increased cortical gyrification in the right inferior frontal and paracentral regions. And these results suggest that a more distinctive allocation of REM sleep over the 24-hour cycle is associated with regional cortical gyrification in aging, and thereby point towards a protective role of circadian REM sleep regulation for age-related changes in brain organization. So aging classically goes along with a decline in sulcus depth, and these results may underline a protective role of preserved or youth-like circadian sleep regulation. And now we move to the microstructural level, where we measured, again, the circadian amplitude of REM sleep through a 40-hour multiple nap protocol. And this time we also measured brain microstructural integrity with quantitative multiparameter mapping in 86 older adults. So here we observed that a reduction in circadian REM amplitude was related to lower magnetization transfer saturation and R1 and R2 star values in several white matter regions, mostly located around the lateral ventricles. But we also observed that it was also related with lower R1 values in gray matter clusters encompassing the hippocampus, the parahippocampus, the thalamus, and the hypothalamus. And R1 has been shown to be primarily impacted by macromolecular content, such as myelin, membrane lipids, and proteins, but also to a lesser extent by iron content. And the hypothalamus is a key structure in sleep-wake regulations, and therefore it might be particularly relevant for understanding the association between circadian sleep regulation processes and brain structure in older adults. In everyday life, the most visible manifestation of altered circadian sleep regulation is an altered temporal organization of the 24-hour rest activity cycles. So the circadian clock is timed to promote wakefulness during the day and sleep during the night, and the circadian alteration would thereby lead to a less distinct allocation of rest or sleep to nighttime and of activity or wakefulness to daytime. And this could lead to fragmented 24-hour rest activity cycles as depicted by the actigraphy recordings on the right, where we can see that for the top one there's a clear distinction between day and night, and while for the bottom one there seems to be the distinction is less clear and there's some rest also during the day. So within this context, it could be assumed that chronic napping in the aged reflects an altered circadian sleep regulation. Napping habits increase with advancing age, such that more than half of adults aged 70 or above nap at least twice a week. And epidemiological studies link chronic napping habits to cognitive decline and also increased beta amyloid burden, especially if naps are frequent and long and unplanned. And studies are starting to use chronic napping in the aged as a health risk factor during aging. So the question is if chronic napping is a reflection of altered circadian sleep regulation, and is chronic napping a health indicator of the aging brain? So to investigate this, we compared 30 nappers to 30 no-nappers. And we can see here on the left that the melatonin secretion over the 40-hour protocol was slightly reduced in the nap group compared to the no-nap group. And this is especially true when the profiles are normalized to the baseline levels, as nappers tend to have higher baseline melatonin levels. For nappers, we also observe an increased sleep efficiency during daytime compared to no-nappers, and also, therefore, a decreased circadian modulation. And we observe also an increased REM an increased REM expression during the day compared to no-nappers. So if we want to look at napping and brain aging, we look at the activity-derived rest activity patterns and NPM maps. So we looked at the fraction of daytime rest of all our participants, derived from actigraphy, and we looked at magnetization transfer saturation in the hypothalamic gray matter, which is important for sleep-wake regulation. And this data is for 78 older adults. And we observe a correlation between the MT set values in the posterior hypothalamic gray matter tissue and daytime rest. So the posterior hypothalamus is wake promoting, and the decrease in the MT value is associated with an increased daytime rest. So the association remains significant after adjusting for nighttime sleep fragmentation and after excluding potential confounding effects due to transition periods during early morning and late evening hours, but also after removing the contribution of the mammillary bodies. Finally, we also use modeling to look at daytime rest, and for the same subjects, we fit this model to the data derived from the lab. So we looked at sleep timing and duration and melatonin onset. And then if we extract the model parameters, we observe a link between the daytime rest, as measured with actimetry in the field, and the model parameters based on lab data. So we observed that the circadian strength derived from lab data is correlated with daytime rest as measured in the field. And note that circadian strength is here parameterized negatively, so actually a decrease in circadian strength is correlated with an increase in daytime rest. And this points towards circadian dysregulation. Now to conclude, when we age, our sleep changes, and there seems to be a contribution of an alteration in circadian drive, and we aim to investigate if age-related changes contribute to cognitive and brain aging. Chronic napping in the age seems to be associated with changes in circadian sleep-wake regulation. However, sleep timing is important. Daytime rest might be a health indicator in the context of healthy aging, but nap characteristics are important as well as reasons underlying napping. Not all naps are the same. And our goal is to be able to go from sleep phenotypes to infer on sleep regulation processes. So this is our team, our PI, Christina Smith on the left, and our collaborators. Thank you.